Nobody told me I'd be following Phil today, so I hope you all stick around. So he talked a lot about characterization of AAV vectors, and I'm hoping to extend that conversation just a little bit, and that's like the what of what's in the particles. I'll talk a little bit about that at the beginning, and then get into a little bit more of the why or the how we're trying to make products that would to make better vectors, essentially, to help us out, and using in silico technologies, in silico vector modeling to help us out. So I usually show this. I think this group is pretty familiar with this, just to state that, you know, when we think about designing a vector, we have all of these combinations in mind when we start out, right? Early research, we're trying to think about promoters, transgenes, polyase, but not just these things. We're trying to think about, you know, introns. Do we include those or not include them, you know, based on what the biological outcomes may be. 5' UTRs, 3' UTRs, all these combinations you want to think about. So how do we expedite that early research? How do we get to a candidate therapeutic, I guess you might say, a little faster than your typical strategy of having to like bombard 1,000 different constructs and try to analyze all these constructs in an early research program. So we at Solid, we've been working closely with Form Bio. That's why I'm here today, and thanks to Mark, he invited me to give a presentation. So we've been working with them, of course, because there's a lot of advantages to that. We have, you know, expansion of expertise. They have the expertise in bioinformatics, software engineering, and data science. So we learn from them. They have the capabilities that we can take advantage of. I guess we're using them a little bit. They're using us too. We're providing them, you know, data to input into their systems so that they can build better systems, essentially. So we have expansion of knowledge and we have expansion of infrastructure by working together. So there's a lot of advantages to machine learning and gene therapy. I'm not going to sit here and talk to you about machine learning. That is not my expertise. That's Form Bio's expertise. I think Amy might speak a little bit to that a little later. But you know, you have the ability to analyze large data sets accurately, identify patterns, and optimize parameters, and then handling these data-intensive tasks is very important, and it's important to work with folks who have the capabilities to do that. So when we think about where are we going to optimize, where are we going to get in early, we really want to do that at the very early stages. So you've heard Phil talk a lot about making AAV, characterizing AAV. You know, this is just an example of a triple transfection on HEK cells. You put the plasmids into the cells. So you really want to get at that early stage where you're designing your vector. You're going to get this heterogeneous population of vectors like Phil just talked to you about. But the goal of that is to really, whether that's in process, whether that's early, where you're designing your vector and you're trying to increase the amount of full-length vector that you're getting packaged into the particles, you want to get to something where your drug product is pure as possible. And then you probably want to do this before you get to, you know, a GMP process or something where you're trying to scale up. So you're going from, you know, a 2-liter process to a 2,000-liter process and making a product that you're trying to get out into patients. So as early as you can possibly get to a candidate that you think will give you the best odds of success in the clinic eventually. So again, going back to the what of molecular species that are in a vector population. So vector characterization, we just heard Phil's great talk on basically just this. And we're taking advantage of a lot of the technologies that he started for the field, basically. So characterization of molecular species in AAV particles, again, that heterogeneous population, what are the key needs? We're looking at, you know, we want reproducible library preparations, reproducible long-read sequencing capability. So are we going to use, you know, Oxford Nanopore? Are we going to use PacBio's long-read sequencing? Why do we use one versus the other? What machine are we going to use, even if you're just doing PacBio? Are you going to use the Revio system? Are you going to use a Sequel IIe? All of these things are important, and when you start changing things along the way, you'll start to see differences, I guarantee it, in the outcome of what you're looking at in the characterization of the species. So we need to get some way, we need to figure out how to make these things reproducible, and we need to probably settle on, you know, instruments, on library preparation processes, on what the annotation is. What are we calling these things? The nomenclature that we're using. So there's a lot of things that we're working on, I think, as a team within this group that Form Bio has put together to get to that. So you want reproducible computation in bioinformatics. That's the pipeline that they've kind of put together. I know there's a lot of independent groups working on different bioinformatics pipelines, but it'd be nice if we can unify that to some extent in the field. And of course, data provenance is really important. So the ability to go back to your data sets, the ability to pull those out whenever you want and have this centralized access and be able to access those. Solutions, doing external or internal routine library preparations, possibly bringing in your own internal sequencing capabilities. If you're working with CROs, from experience, I'll tell you, be careful and make sure you have vast insight into what they're doing, or try to have insight into exactly what they're doing in their processes. You want to work with an established bioinformatics pipeline, and then of course cloud access for immediate secure data access and chain of provenance. So we have we're working with Form Bio, like I said, and PacBio sequencing, and we're working with various CROs to have an end-to-end, basically, workflow that includes, we prepare the vectors, we have our own purification, our own vector production in-house, and then we basically ship everything out at various CROs to help us with the vector purification, the thermal annealing that Phil mentioned, and then doing smart bell adapters and doing the sequencing. We have typically used a Sequel IIe system from PacBio, and then we do all of the bioinformatics and get the results out. This is just an example from Form Bio of some of the bioinformatics that you get out. This is a couple cartoons from single-stranded AAV subtypes versus self-comp AAV subtypes. They're very similar. You get partials, a lot of things that Phil already spoke about. But you also have these things that I'm going to talk about in a minute. So when you're looking for, and Phil mentioned it too, when you're looking for single-stranded AAVs, you're going to get some of these snapback genomes, and some of the nomenclature we've been using, we still call these self-comp. They're not really self-comp because self-comp is something you intentionally design and develop. So they're really these snapback genome structures that occur in your preps, and I'll show you some data here on that. So this is some of our data from our in-house work and working with Form Bio. If you look to the left, this is more of a general capture of the kind of different forms, the molecular profile that you would get, where you look at single-stranded AAVs, you get these snapback genomes, which often we've been referring as self-comp. They're not self-comp, but they're these snapback genomes. You can see that's a high percentage in this case with this vector packaged into these capsid particles. We have this category that they have called other, still working out the characterization of what some of those molecular species are and how to bin those so that we can improve the annotations. We have host cell DNA, as Phil mentioned, rep-cap, add helper, so often you'll have add helper packaged into these, chimeric vector forms, as well as what's characterized basically as unmapped sequences. But if we take that single-stranded AAV profile, if you were to stop there and you were to say, oh, okay, single-stranded AAV, I got all these full-length vectors because this profile comes back and says, oh, it's single-stranded AAV. But if you break that down even further, if you take a deeper dive into that, you actually will see what the full length versus the left partials, right partials, partials being things that you just have in the middle, basically, of your vector, and then vector plus backbone. And there's many other forms, but these are the ones I'm showing you here are the major ones. You can see that you really go from, like, in this case, 73% of single-stranded AAV down to 50%, for instance, of your full lengths that are actually there, and that there's actually a lot of right partials and left partials. And I'm going to show you how we can use some modeling to help us predict, and then how we can actually change those proportions so we can go from full sequence percentages that are 50%, if you keep that in mind, and bring that up by changing some of the vector elements, basically. So one of the other nice features in working with Form Bio is that they can profile all these structures, similar to, you can use IGV like Phil showed. That's great. I actually really like IGV also. But then they also profile these in their own cartoons in their own way against the consensus sequence. And you can see where these sequences begin to stack up along the consensus sequence. And it gives you an idea then of where, okay, left partials, they're all stacking up here. So there must be a sequence I can go look at that particular site, and I might be able to modify that particular sequence to get me to a place where I'm packaging more fulls. The other thing is that the host genome analysis or characterization feature, I think, is excellent. I'm only showing you the chromosomes, but it actually gives you can actually identify that if there's specific genes that are involved, it'll identify specific genes and tell you which genes you may have a higher proportion of being packaged in regions that are being packaged into your particles. Okay, so that was the kind of the characterization or the what. I want to talk a little bit now about the why. So understanding why you have it, and then taking the why, changing it, and trying to make a better product. So we go back to the challenges. We have vector design issues that lead to molecular subspecies, like I just showed you. We want to identify genetic elements that may potentially lead to these truncations, elements that lead to snapback genomes, putative CPG islands. So things that may affect some of your downstream phenotypes when you start to go into your preclinical studies. So how do you use in silico modeling to do this, to basically reduce, identify, and then get to a point where you can reduce or eliminate some of these truncations, optimize your yields, and then use CPG modeling. This is just a cartoon, really, of a lot of secondary structures, tertiary structures, various elements that you might find in a construct. You might inadvertently add them to it, or you might intentionally add them to it. If you're doing short hairpin structures or something like that, you might be trying to express those short hairpins or put them in the context of an intron, depending on what your therapeutic is. If you're doing that, though, I can guarantee you, unless you're making certain modifications, you will probably get a truncation, or a lot of your product will be truncated, and your full-length product that you're after will be reduced. So what does the in silico AI workflow look like? So this is all Form Bios. This is their Form site, AI foundational model. You hear more about that hopefully later. To me, I call it the black box. Something goes in, something comes out that you hope is better. So you have a bunch of public and proprietary databases that they're using, and then you have private information that can also go into this. And then what you're getting out is hopefully a better construct that has reduced truncations, CPG islands, like I mentioned, and then will lead to a better product that gives you better expression, hopefully something that you can eventually predict. So what does some of these in silico designs and optimizations look like? And this is an example of basically one of the in silico modelings. This is a construct. I guess I better use this. This is a construct here, shown at the bottom, and then across that you basically have the forward and the reverse strands. So the forward strands here and the reverse strands here. And what you'll notice right away is that you can even have, this is a truncation propensity prediction, so where you might have truncations throughout that structure. And it gives you kind of an idea of these various regions where truncations might be occurring. So what becomes pretty evident here, and has been in some of the work we've done, is that those truncation propensities can differ whether you're looking at the forward strand or the reverse strand, which is interesting in itself. But by knowing this, you're in the CAG, you're in the CAG promoter region here, you have a lot of stuff that's going on here, secondary structures going on here. How do I, it's a promoter, so it makes it kind of challenging. You're not going to just go in and start mucking with nucleotides. So, do you swap it? Do you change it out? Do you modify it in some way to get you to a point where you're going to have a better vector? The other thing you can look at are secondary structures. So this is the same construct basically, but this would be the promoter region where you're seeing a lot of secondary and tertiary structure. Overlapping that, on top of that, the truncation prediction. So it gives you an idea where to look within the context of your vector. And then you have GC percent across the vector in blue and orange. You have CPG islands, or CP gives you high, where you have high percentages of CPGs, and those are predictions, very much predictions, not, you know, there's the correlation we don't know with the biological outcome yet, but predictions that these would be regions of high GC interest that you may want to reduce, so CPG reductions. So one of the things you could do here is you can rapidly evaluate multiple, you know, essentially, eventually millions of combinations, right, in silico, and to get to what would eventually be an optimized construct. So this is an example, one of our in-house examples. So 5 different vectors I'm showing you here, 5 different promoter elements that were swapped out from this top one. You can see this is the, I think everything's showing up. Yeah. You can see this is the prediction. These are the secondary structure predictions. When we changed out the promoters, you can see that a lot of this in the prediction models goes away in this region, so in the promoter region. When we look at the real-life data of that being packaged into the vector particle populations, it's very interesting. So similar to what I showed you earlier, this is just the single-stranded AAVs, and then the snapback genomes. You can start to see that there's differences that fall out with vector 1 versus the others here. When you take this population and you break it out a little further into full length or partials, you can see that there is definitely a difference where you're picking up greater than 25% increase in the full lengths that you're packaging versus the right partials, which are decreasing by about an equivalent amount. So you've basically impacted your vector product here quite a bit by changing out those promoters. This is a summary table of what I just told you, essentially. So these are the percent predicted truncations, the percent full length, which is really just adding to 100% essentially. But then this is the real life values here. If you look at vector 1 here versus these, you'll see that the trends, the numbers aren't exactly a match, but the trends are very similar in the predicted versus the vectors, the outcome of the vectors. So it's important that I note here that this is great. This is all fine and dandy. It's nice. This is where you start. You start with the vectors being packaged into the particles, and you start with production, and it gives you an idea of how much full length you have. But then you have to be able to say, okay, does this translate into our preclinical models, and then obviously into our patients? So you run it through this. You know, you're going to run it through some iterative process that's going to get you through preclinical models, whatever they may be. If it's not, then you're going to go back through, you know, to redesign your vectors. Hopefully, you've done good enough design up front, or you've had some good designs up front that get you through your preclinical, and that get, you know, and expedite that process, and then eventually get you into your IND and in the clinic. I think I was pretty quick. So in silico tools, you know, these partnerships are instrumental to understanding the quality of your drug product and accelerant for improving these ultimate designs. So thank you all for your time.